There we go. Okay, so yesterday we were talking about the endomembrane system. So I left this drawing up here from 6th hour yesterday when I drew with that class. So remember the endomembrane system. The membranes are either physically connected or are connected via a transport vesicle. So this was the nucleus and the nuclear envelope with the rough ER, protein made, buds off the rough ER, and the transport vesicle travels to the Golgi apparatus where it fuses with that membrane. The polypeptide goes through and is modified, buds off again, and becomes another transport vesicle that fuses with the cell membrane, and the protein is secreted from the cell. And I gave the example of that happening with your stomach yesterday. So what I want to do is talk a little bit about, I'm going to add this on to it, the ribosome. And so the ribosome, remember we drew a ribosome yesterday attached to the rough ER. But ribosomes can be what are called free ribosomes, where they're not attached to the rough ER, or bound, which they are. So some ribosomes are floating out here by themselves. We talked about the ribosomes yesterday, that they both manufacture proteins. They both make proteins. The difference is that the bound ribosome, like we talked about yesterday, makes proteins that leave the cell, that are secreted from the cell. Ribosomes that are in the cell are floating around by themselves in the cell, make proteins that stay in the cell. So they will be used in the cell. So it might be an enzyme that's used in the cell, and so on. So that's the difference between there, and I didn't quite talk a little bit about that difference yesterday, and I want to point that out today. All right. So the next thing is the lysosomes, and the lysosomes go into this this picture here. So what we talked about yesterday in the making of the transport vesicles, this is one pathway that this buds off the Golgi apparatus, it can become a transport vesicle and fuse with the cell membrane, and it leaves the cell. There's another pathway that you can go, and that is that instead of it becoming a transport vesicle that fuses with the cell membrane, this can become, I'm going to put an arrow down here, this can become a lysosome. Did I draw this yesterday for you? No. Okay. This lysosome, we're going to write here, stays in the cell. It's an organelle that's found in the cell. So it's made in this process, and I'm going to draw the little polypeptide. So this, in a lysosome, these are enzymes. Because remember, enzymes are proteins. So instead of the protein being secreted from the cell, this little guy is going to stay in, and it contains enzymes. I'm going to call that pathway number 2. So that's a second thing that could happen to this that's budded off from the Golgi apparatus. So if it becomes a lysosome, stays in the cell, has enzymes, what does it do? So let's write here in our notes. So a lysosome is a membranous sac of enzymes, so we just wrote here, that can digest macromolecules, so proteins, carbohydrates, lipids. They work best in an acidic environment. So inside the lysosome is very acidic, and that's because, I'm going to put here acidic environment here, and that's because that protein, remember proteins are very sensitive to pH. Most proteins in our body work best, and enzymes work best in a more neutral pH, but there are certain ones that work best in a more acidic pH. And so in a cell, a lysosome has acid in it, and that's because that's the pH that those enzymes work the best at. That's also why, like in our stomach, we talked about our stomach yesterday, enzymes actually digest your food. So remember we talked about a chain of proteins, and the enzymes digest that protein that you've eaten by breaking the bonds. This enzyme actually does the digestion, but the acid actually provides an environment for those enzymes to work. So those enzymes actually work the best in an acidic environment. So the acid is contained solely in the stomach.
Can you explain like the nucleus part? Like I'm way lost because I forgot. I didn't think.
Like, just explain what.
Yeah, like what's going on with the amino acids through the.
Okay, so go over this part again.
Yeah.
Okay. So the nucleus I put on here just to show that the double membrane of the nucleus is continuous with the membrane of the rough ER.
Okay.
All right, so it's connected there. And so then you also have the ribosome that's attached to the rough ER. And the ribosomes make proteins, and proteins are chains of amino acids. So I put just AA standing for amino acids connected together, trying to illustrate that at the ribosome is hooking together those amino acids.
Okay.
So then you get a finished polypeptide. All right, so it's the amino acid chain is built, and then that buds off of the rough ER through a transport vesicle and goes to the Golgi apparatus.
Okay.
All right, does that make sense?
Yeah, that makes sense. Thank you.
Yep. All right, so the enzymes in the lysosomal membranes this is made by the rough ER and transferred to the Golgi apparatus. So that's what I was showing here, that this is how these are made. All right, and so that last bullet is adding this picture on here showing that the lysosomes are made by that process. Am I good? Too fast? Oh, yeah. Okay. So I'm going to give you some examples of the job of the lysosome. So what do these enzymes do? They can digest stuff, but in what situation would they do that? So I'm going to just write a few bullets here that I'm going to show you in a picture here. So some types of cells can engulf another cell by what we call phagocytosis. And I'll illustrate that in just a minute. And this forms what we call a food vacuole. Then the lysosome fuses with the food vacuole, digests the molecules. So that's job number one. Job number two is that they can also use the enzymes to recycle the cell's own organelles and macromolecules. This is called autophagy. So an example of this is in development for a frog, so the tail of a tadpole. The tail goes away. How does it do that? They are digested, but with the help of the lysosomes in that cell. And this one, is there an empty blank?
Yeah.
Okay, yeah. And so the empty blank is when lysosomes kind of destroy cells. That's called cell suicide, but the name, the proper name is called apoptosis. APOPTOSIS. So it's kind of like programmed cell death, okay? And so that happens a lot in development. So this is an example. It happens in us in development. When we're developing in our mother's womb, there is actual cells between our fingers and our toes. And during development, those cells undergo apoptosis, and we kill them on purpose so that we can have movable fingers and toes. All right, and so that's an example that the lysosomes play a role in that, in killing the cells. Okay, so here is the pictures of the two basic things that the lysosome can do. So let me explain what you're looking at here. This usually happens in unicellular organisms. So I'm going to put here in unicellular organisms, meaning that they're only one cell big. And if they're one cell big, they have to get food just like you and I. And if they aren't an organism that has chloroplasts that can make their own food and do photosynthesis, they have to take it in from their environment. If you're only one cell big, you don't have a mouth, you don't have a digestive system, and so on. And so how does this one cell big organism get its food? This is how it does, through this process called phagocytosis. What happens is, here's my unicellular organism. All right, I'm just going to draw a circle for the unicellular organism, and I'm going to put a dot on the outside. That's its food. So it somehow has to get the food inside the cell. The food, though, typically is too big to get through the cell membrane, so it's not squeezing through the cell membrane. So it has to get in another way. So what this unicellular organism does is begin to indent its membrane around that food. So you see that indentation around it. And it keeps on indenting and surrounding that, and eventually, well, I guess I'll just draw another picture here. Eventually, it's going to look like this, with the food inside of it. And this is going to continue, I'm going to put in red, that this is going to continue to pinch off until eventually you get what's called a food vacuole. Because why? Because it contains the food that it tried to get in. So this is your food vacuole. So now we've got its food in. That's like us chewing and swallowing. All right, that's how we get our food in. But we can't use our food, even though we chew it, it breaks down into smaller pieces, but it's not digested. It's still a protein, it's still a chain of amino acids. We need to break the bonds between them. So this food still needs to be digested. This little guy does not have a digestive system. So this right here is what you're looking at right here. So back to looking at this picture here, so this food vacuole is right here. So this is where it's coming in. So that's why I want to show you the big picture of what you're seeing here in this picture. So here's your food vacuole, that's this one here. What does the digestion? In comes the lysosome. Lysosome is something that's found already in the cell that has digestive enzymes. What does the lysosome do? It fuses with the vacuole. Those enzymes mix with the food, and the food is digested. And then the food, when it's digested, is in small enough molecules, then can diffuse out of the vacuole and be used by the cell. All right, and so these guys here, this is the smaller molecules, can diffuse out and be used by the cell. So that's how a small organism gets its food. Example number 2 of what a lysosome does is this autophagy. It engulfs, so here's the lysosome. Vacuoles kind of surround old, damaged organelles. So a peroxisome is an organelle. This is not on our list of what you need to know for this class. It's just know that it's an organelle in the cell. And mitochondria is an organelle that you're going to need to know about. We'll talk about later on this hour. Contained in a vesicle. So these guys are damaged, and so we don't want damaged organelles to be floating around and being used in the cell. So what happens is the lysosome fuses with this vesicle and destroys basically the old damaged organelles. So that is another use of the lysosome. Okay? And so I just wanted to also show you that when any of these organelles, which is kind of what your homework was the other night, is to what kinds of things would happen if you didn't have a functioning certain organelle. So this is the case for a lot of diseases. A lot of diseases that people can get can be because their organelles, an organelle is not functioning properly. So for this one here, Tay-Sachs disease is a disease where the lysosome inside the enzyme is not made properly. And this enzyme is supposed to break down lipids. And so that's a natural thing that happens. And so people with Tay-Sachs disease have an abnormal enzyme in their lysosomes. So their lysosomes can't do exactly what it's supposed to do. And so it's supposed to break down those lipids. Those lipids don't get broke down, and they get an accumulation of lipids in the brain and impedes the brain function and so on. So, and that's all because of one abnormal enzyme in the lysosome. All right, so that's an example. There are a lot of, we'll talk about the mitochondria in just a little bit. There are a lot of, tons of diseases where people have something abnormal with their mitochondria, all right, and so on and so forth. So it can cause a lot of different diseases. All right, we're going to add on to this picture in just a second here. Another thing that can be made is the vacuole. They're large vesicles derived also from the ER and the Golgi apparatus. So let's add on to this. So I have here pathway number 1, pathway number 2. Let's add pathway number 3 here. So this, the third thing that could go on here is that this budding off of the Golgi apparatus can become a vacuole. This also stays in the cell. And let's put my number 3 here. Okay, so that's the third pathway, the vacuole. And I want to point out, like the lysosome, a transport vesicle, whether it be this transport vesicle or this one, and the vacuole. Just say, oh, I made them all circles. Like, so if you looked at a cell underneath the microscope, you wouldn't be able to tell the difference between them, all right? They look the same. They're just small little circular organelles. And so there are different kinds of vacuoles that perform different functions. We just talked about one in the unicellular organism, the food vacuole. So those guys are formed from phagocytosis. Another one we haven't talked about is the contractile vacuole. Found in many freshwater protists. So let me talk about what a protist is here. So freshwater protist, this is a unicellular prokaryotic cell. What does it mean to be a prokaryote? No nucleus or not necessarily a cell wall, but membrane-bound organelles. All right, so mitochondria, chloroplasts, endoplasmic reticulum, all of that kind of stuff. It says it pumps excess water out of cells. So what happens is these guys are found in fresh water. So when you go swimming in a lake, you're swimming with protists usually, all right? So they're unicellular organisms. Let me explain here. The paramecium kind of has a shape like this. So it lives in fresh water, and what happens is the water will diffuse inside the paramecium. This is a, what will happen then is that when the water goes in and kind of fills up, kind of like a, I'll use the analogy of a water balloon. So it kind of fills up. What happens is it runs the risk of exploding because too much water goes in. So what happens is there's this what's called a contractile vacuole that collects the water. So I'm going to add an arrow that shows that's going into the contractile vacuole. Then as that contractile vacuole fills up, it kind of goes to the surface here, all right, to the membrane, and it contracts. It actually squeezes and actually forces the water back out. So what happens is that that little guy will never die because it's going to explode of water, all right, when water goes into it. And I just made it's a eukaryotic cell, sorry. Eukaryote. Let's change that. Just realized I wrote it wrong. And so we don't have contractile vacuoles. Our cells do not have contractile vacuoles, but usually they're unicellular organisms that run that risk. The third and last type of vacuole is a central vacuole. This is found in plant cells, And they store a lot of things. They store organic compounds, pigments, poisonous compounds, and water. And so what happens is when we look at a plant cell, so here's the cell wall of a plant, and then right underneath the cell wall, what do plants have? What do you think? It's underneath the cell wall. Right now? Cell membrane. They have two because they have different functions. The cell wall is for structure. The cell membrane regulates what goes in and out of the cell. Then, when we look at the plant cell, there's a huge part here that it's a huge circle. So I drew a vacuole very small here with plants. They have this specialized vacuole. This is the central vacuole. It's huge in the plant. So much so that usually when you look at plant cells underneath the microscope, that the chloroplasts and all the other organelles are like squished up against the cell wall. They're like squished up because the central vacuole takes up so much room. And so what does it do? It holds water. So when we water plants, and water goes up through the roots and up into all the cells of the plant, the water, when it goes into the cell, is stored in the central vacuole. So what happens is that the central vacuole actually helps to give plants its structure along with the cell wall, because the central vacuole, when it fills up with water, do you see that it's kind of will balloon up and put pressure on the cell wall, and it makes the cells stand up nice and straight, all right? That's why if you don't water plants, what's the first thing that happens to the plant usually? It wilts, right? And so that's the reason why, because so that it's not getting any new water. So what is the plant doing? It's relying on the water in these central vacuoles, and they're taking it out, which means that then there's not as much pressure, and all of those cells will wilt. All right, so that's your central vacuole, but it doesn't just contain water. So it says pigments. So when we look at a flower, let's say you look at a red rose. The petals are made up of plant cells, and in the central vacuole of those cells making up the petals are a red pigment that gives it its red color. All right, so that's what it means, the pigments here. Poisonous plants will store their poisonous compound in the central vacuole as well. So it's a storage of many different things. And so that's unique. Animal cells have vacuoles, but our vacuoles are much smaller, like I drew here. Plant cells has this unique central vacuole. We don't have a big central vacuole. And so this is what I was saying here. It takes up a good portion of the cell. All right, so that rounds up the endomembrane system. All right, so what you should be able to do is, like looking at this drawing here, you should be able to explain something like this. So if I asked you to describe how a protein is secreted from the cell, you should be able to start off with saying a bound ribosome to the rough ER puts amino acids together to make a protein. That protein buds off of the ER into a transport vesicle that travels to the Golgi apparatus, that moves, the transport vesicle fuses with the membrane of the Golgi apparatus, and that polypeptide is modified through the Golgi apparatus. When it's done being modified, it buds off of the membrane from the Golgi apparatus, becomes a transport vesicle that fuses with the cell membrane and is secreted from the cell. Okay? So that's something that you should be able to, that's a big thing for this chapter.
So the ribosomes are those, is that polypeptide already made, or is that made?
It's made at the ribosome. The ribosome is where it actually attaches the amino acids, and then as it grows, the part that's already made kind of hangs out in the rough ER until it eventually detaches from the ribosome, and the whole thing is in the rough ER.
Okay.
All right. Okay. And then similarly for a lysosome and a vacuole, all right? And then knowing that proteins that stay in the cell are made just on the free ribosome. So that's something that you should be able to do, all right? Okay, so that is the endomembrane system. This was a big new part, I think, that was different than biology in terms of how all of these organelles work together in one system. All right, let's look at two other organelles that I know you are familiar with, mitochondria and the chloroplast. Everybody always knows the mitochondria and refers it to as a catchphrase usually of what the mitochondria does. What's that?
The powerhouse.
The powerhouse of the cell. All right, so what does that mean? Grayson. It makes ATP, which is used for energy. So that's what we need to know, okay? So you know, we wouldn't write the powerhouse of the cell. You can think it to help you to remember what it does, but we need to be a little bit more specific about that. So we're going to look at the mitochondria in the chloroplasts first to see what things make them unique compared to all the other organelles that we've talked about. We talked about that yesterday. What does the mitochondria and the chloroplast contain that the other ones do not? We talked about that yesterday. Go ahead. Go ahead. DNA. So they have DNA. All these guys, other ones, do not have DNA. There's no DNA in there, and so that makes them unique. And so, we're going to talk about some theories about why they have DNA and the other organelles do not. So mitochondria, the cytocellular respiration, uses oxygen to generate ATP. And this is generate ATP used to do cellular work. So this is the energy source for. And let me clear up maybe some confusion, because when we talked about monosaccharides like glucose, we said glucose is used for energy from the cell. How does glucose fit into what goes on here in the mitochondria? How does glucose fit into this mix? Because I just said that ATP is used to do cellular work. So ATP is actually the direct energy source to do work in the cell. What's the relationship between that and glucose? Want to try it, Grayson?
The glucose goes to the mitochondria and breaks it down.
Yes. So when it says that a metabolic process that uses oxygen, I'm going to add here uses oxygen to break down glucose to provide energy to generate ATP. So that phrase could go in between there. And so without glucose, you don't make ATP, and then you don't have cellular work. So glucose, that's why we say glucose is used for fuel or energy. Chloroplast, on the other hand, is the site of photosynthesis. Photosynthesis uses not oxygen, but carbon dioxide and light, and also water to make glucose, all right? And so let's look at each one, at these relationships between them. Did I go too fast? Go back. There's some scuttling. So mitochondria and chloroplasts actually have similarities with bacteria. So bacteria are considered their own organism, right? They're prokaryotic cells, they're free living, and the similarities are that mitochondria, chloroplasts, and bacteria have a double membrane that they're surrounded. All of them do. They contain free ribosomes and, as we said, circular DNA molecules. Bacteria don't have any other organelles. They just have DNA and ribosomes and then the cytoplasm. And the thing is they have everything that they need, because DNA contains instructions to make proteins. Proteins are made at the ribosome, so they can make their proteins, so they have all their enzymes and so on, and so they can live just fine. And so mitochondria and chloroplasts actually have that same thing that the bacteria do. And then also they grow and reproduce independently, meaning that bacteria can grow and divide and make new bacteria. But guess what? So can our mitochondria and chloroplasts, or in plant cells and chloroplasts. Mitochondria can divide and make more mitochondria. And so that's also similar to the bacteria. And so this woman named Lynn Margulis came up with this theory called the endosymbiont theory, or it's called endosymbiosis. And her theory was that, basically, long story short, her theory is that because the mitochondria have all these things in common with bacteria, and bacteria are free-living organisms, her theory is that the mitochondria and the chloroplasts, way back in the day, were also once free-living organisms like bacteria. All right, that's what's her theory. So we're going to write about her theory, and then I'm going to show it to you in a picture. So her theory suggests that an early ancestor of eukaryotes engulfed an oxygen-using, non-photosynthetic prokaryotic cell. And that formed a relationship with the host cell, becoming an endosymbiont. And these are what became the mitochondria inside the host cell. Then something similar happened with the chloroplasts. At least one of these cells may have then taken up a photosynthetic prokaryote, so a free-living organism that does photosynthesis, which then became part of the cell and became what we now know as the chloroplast. So that's what this picture is trying to show you. So here is your ancestor. Did I go too fast again? Good. All right. So here's the ancestor of the eukaryotic cell. So here's the nucleus of the double membrane, endoplasmic reticulum. And this picture is showing you, it says engulfing of oxygen using, so this uses oxygen but can't do photosynthesis, kind of like the mitochondria, prokaryote, so it was once free living, which then was engulfed. Notice here it's engulfing it much like the unicellular organism was engulfing its food that we looked at earlier. So it engulfs it and pinches it off, and now it becomes part of the cell. This mitochondria, the cell that's now part of it became the mitochondria, which makes ATP. It was good for the cell, and so therefore became an organelle. All right, so now we have the mitochondria, and then the second part of that is that if one of these guys who did this took up a free-living photosynthetic organism, like this little guy here can do photosynthesis, engulfed it the same way, indents the membrane, pinches it off, and now the cell can make its own glucose and break it down to do ATP. That's a good thing, and it became part of the cell, and that's the chloroplast. And so that's her theory, that they were once free-living organisms, and that theory is based upon the fact that the mitochondria and chloroplasts are like prokaryotic cells. All right? So, so let's look at then more directly about the mitochondria and the chloroplasts. So we wrote that the mitochondria does cellular respiration. It's found in nearly all eukaryotic cells to make ATP. Let's talk about its structure for a second. So it has a smooth outer membrane and an inner membrane folded into cristae. So I'm going to do a little picture here. Mitochondria is kind of shaped like a bean, a little bit. This is the outer membrane. Outer membrane's smooth. Inner membrane, so there's a membrane inside, is folded. So this is the folds like this. So this is the outer membrane, inner membrane. Inner membrane folded. What is it? Why do you think that inner membrane is folded? To increase the surface area. That's exactly right. Usually, when in biology, remember, structure determines function. Usually, if you see folds or a lot, then that usually means that it's there to increase surface area, which means that that structure is probably pretty important to what goes on in the mitochondria. So what goes on in the mitochondria is we make what? ATP, right? And there are enzymes that make ATP. So if I make a little key here, if every one of these red little dots is an enzyme to make ATP, And the enzymes, guess where they're located? They're embedded in the membrane. Remember when we did the cell membrane, you have the lipid bilayer, but you also have proteins embedded in the membrane? These enzymes are actually embedded in the membrane. So therefore, these enzymes all are in these folds here, and these enzymes speed up the making of ATP. So guess what? The more inner membrane you have, the more enzymes you have, which the more ATP you have, which then the more cellular work you can do.
So when you said it increases the surface area, does that fold eventually like unfold and then it increases? Is that what you're saying?
No, it doesn't have to. It doesn't have to. So it doesn't unfold because it's contained within this outer membrane as well. But what folding does is let it increase the surface area without having to increase volume of the space. Does that make sense?
Yeah.
All right. And so, so that's what actually happens there. All right. So the inner membrane creates two compartments. So we have a compartment in between the membranes called the intermembrane space, and then we have the mitochondrial matrix. So inside here is the matrix, right here. And so a lot of the steps for cellular respiration occur in the matrix, but the actual making of the ATP is with those enzymes in the cristae or in the folds. So the cristae, the folds, present a large surface area for enzymes that synthesize ATP. So that's what I try to draw there. So this is just another picture of the mitochondria. This is showing you the cross section. This added two other things that I didn't put into my drawing here. It's just illustrating DNA. So this right here is the circular DNA molecule and the free ribosomes. So all these little dots are ribosomes. So therefore, again, could have been a free living organism at one time. And that's the mitochondria. So the chloroplast does the opposite reaction, which is photosynthesis. It captures light energy. And how does it do that? If you recall, they have a pigment called chlorophyll. Remember that? Pigments, what they do is they absorb certain wavelengths of light. So the chloroplast contain chlorophyll, which to our eyes appears green. It has all the other enzymes and stuff that's going to be needed for photosynthesis. So chloroplasts are found heavily in cells that are green, and so because they have so much chlorophyll in them. And so the leaves, a little bit of the stem, most of the photosynthesis occurs in the leaves of the plant. All right, algae as well. So let's look at this chloroplast here. So we're looking at a cross section. So if you notice, the chloroplast has a double membrane like the mitochondria, but these membranes aren't what? Folded. There's no fold in there. Do you see how both the double membrane is smooth? So what that tells me, those membranes have nothing to do with photosynthesis. All right, so they have nothing to do with photosynthesis. What does have something to do with photosynthesis is what there's a lot of. What are there a lot of in this? There are a lot of these little discs called thylakoids. Now, if we look at each one of these discs, this right here, cut it open so you can see that they're hollow inside. Each disc has a membrane around it. All right, and so since there's a lot of discs, there's a lot of membrane, and guess what? That's the membrane that's really important for photosynthesis. And we just said that chlorophyll, it is needed to capture light energy. Photosynthesis can't happen without light energy. So what I'm gonna draw in here is why this membrane and why there's so many of them. You can see here all these stacks of these thylakoids is because within there, I'm gonna draw here in green. So my little green dot here is a chlorophyll molecule. It's a pigment that captures light energy. And so the more thylakoids you have, the more membrane you have, the more chlorophyll you have, the more light you can absorb, and the more photosynthesis happens. And so I'm not going to do it on all of them, but there's tons of chlorophyll. The stack of them, So you can see there are stacks, kind of like poker chips a little bit, all right? The stacks are called granum. I want to point out, sometimes you'll see the word granum. That's the singular form of the word. Grana is plural. We're talking about the same thing here. These stacks of thylakoids, so I just want to point that out. If you see it written either way, you're not confused about what that means. Okay? And so it has the ribosomes, and the stroma is just the liquid inside of it, inside of the chloroplast. So for mitochondria and chloroplasts, knowing what happens there for this chapter is important, but also the structure is important. So one thing to take away from this is how the structure of the mitochondria and chloroplast matches the function. And so that is with the surface area of the inner membrane as well as the thylakoid. So that's a big overarching thing that you need to understand about those. So let's just write about these structures that we just looked at in the picture of the chloroplast. So thylakoids stack up to form the granum, which is the stack. Stroma is that fluid inside. And the chloroplast is one organelle of a larger group called plastids. But right now you just need to know the chloroplast. All right. Cytoskeleton, cyto in front of something. You guys know what that means, in front of something? Refers to the cell. So it's kind of like a cell skeleton. So when we look at a cell, it's filled with, if this is the nucleus and it has the organelles, it has the mitochondria and everything like that. And it's filled with fluid. And while that fluid that it's filled with helps to give it somewhat of a shape, there is a small little cellular kind of skeleton. It's not made out of bones like our skeleton. It's made out of tubes and fibers. So the cytoskeleton is a network of fibers throughout the cytoplasm, and it helps to organize the cell's structures and activities, anchor some organelles, and there's three structures. There's what we call microtubules, microfilaments, and intermediate filaments. Let me talk about these words here. Micro means small, so these guys are small tubes. So really, they're microscopic tubes. These guys are small fibers. They're very thin. Intermediate means that it's not quite as small as micro. What's going on?
The first bullet has 2 blanks that are not there.
Oh, really?
Yeah.
How is it that... Just close my mouth. All right. I'm going to give them shape and structures to the cell. All right. Yeah. No, nothing. All right. So sorry. Intermediate filaments are kind of an in-between size. They're not large, but they're not micro either in their fibers. So they're a little thicker. So I'm just going to draw them a little thicker here than the smaller fibers. All right, that's all that means. So how does this relate to the cell? How does it give it structure? So what happens is, and you can't see this, these are so small that you can't really see it underneath our microscopes, but throughout the cell, here's some tubes. They go throughout the cell. So here would be another tube coming this way, all right, and so on. And then you might have some filaments like this going throughout the cell. And what happens is all of these guys, these thread-like guys here, they go out throughout the whole cell, and it's like a scaffolding system. So it helps to give shape to the cell, all right? And that's what that means. So cell structures. And then another cool thing that it does, relating back here to transport vesicles. So you may have wondered, how does the transport vesicle get from here to here? You were wondering that, right? All right, so how does it get there? It actually can ride on these microtubules, kind of like a little subway system. So it actually scoots along them, all right? And it can go from one cell to another. That's what this next picture is showing you here. Here's the little vesicle, like a transport vesicle, they have this little motor protein that they use ATP to contract, and it moves along these microtubules. And so you have to imagine that that's how the transport vesicle moves throughout the cell. All right. So so that's another purpose of the microtubules. Another purpose of the microtubules has to do with cell division. In animal cells, microtubules grow out from a centrosome near the nucleus, and it's involved in cell division. The centrosome has a pair of centrioles made out of microtubules. And that's what this picture here is showing you. So this picture here, the whole thing is called, this part, and I don't know if you can you see the yellow part in yours? Kind of, very faintly. The whole thing is called a centrosome. These little guys are centrioles. You guys have probably seen them, if you remember from biology, this you'll probably remember, during cell division. So when cell division occurs, when it's preparing for cell division, the cell has a centrosome region, and I'm just going to put these guys darker spots as these are the centrioles. That's this guy here. This guy is the centrosome. When it gets ready for cell division, it divides and makes another whole centrosome, and that centrosome goes on the opposite sides of the side of the cell. So I'm only going to label it there. And then guess what grows from the centrioles? You guys remember the spindle fiber during cell division? Does this ring a bell? All right, it comes out of the centrioles, and the chromosomes line up down the center of the cell, connected to the spindle fibers. That right there, these guys are microtubules. So they help with cell division, with lining up the chromosomes, and so on. So that's another function of the microtubule. All right, I'm going to stop here because I want to talk a little quickly about your homework. The stapled packet that you just picked up is homework. Notice underneath number 1 it says use the AP formula sheet. That's sheet number 2 that you just picked up. This formula sheet is the sheet that you will get on the AP test. So these are all formulas that we'll talk about within the class. If you look at the back side of this formula sheet, on the bottom left are all the formulas that have to do with surface area and volume. That's what you're going to do in the packet. If you look at the second page of the packet, second page, the front side, if you look at the bottom right, it says right cylinder. Cross the right cylinder out. You don't have to do the right cylinder. Alright. Let me explain the back side real quick. Back side has a picture of the endomembrane system. What I want you to do is on this picture down below is I want you to look at the picture. Remember I said that this is what we just went over, basically. That you should be able to explain it. So notice here there's a pathway, there's an arrow. What I'd like you to do is number the number, like so at the rough ER, write a number 1, and then write number 1 down here and tell me what's happening at the rough ER. Then, so you're going to make steps, and you're going to explain it. So I want you to see if you can explain what's happening in this picture in your own words. Okay? All right? Okay, that's it. You guys have any